be Akinori Hishiy, Chief Science Officer at SOLA, presenting preclinical proof of concept of SOL-257, a gene therapy targeting misfolded TDP-43 in ALS. Welcome.
Thank you so much for having me here today. And I'm very honored to have this great opportunity to present and introduce our therapeutics. So I'm sharing this slide, but can you see? Great. We have been developing the protein folding based therapies for many neurodegenerative diseases, such as ALS. And the molecular pathology of these neurodegenerative diseases are fairly diverse from one disease to another. But there's one thing which is these diseases are caused with protein folding issues of some particular proteins. For instance, the Alzheimer's disease is characterized by a senile plaque, of which major component is aggregated amyloid beta peptide, and the neurofibril tangles, in which the aggregated and hyperphosphorylated tau protein is observed. So currently, it is believed that these disease-causing proteins, specific proteins, are fairly prone to misfolding, and at some point, they start misfolding, leading to a neurodegeneration. So simple, I don't say simple, but the one with the therapeutics is just to remove or eliminate this kind of disease-causing proteins. And currently, we have a few modalities, such as ASO, microRNA, and PROTAC-OTAC. ASO and microRNA stops the protein expression at the RNA levels, and the PROTAC-OTAC accelerates degradation of engaged proteins. But we also know that the patients have two types of proteins. The one is good one, which is properly folded, therefore it's functional. However, another one is misfolded, therefore it's toxic to cause disease. And in general, these modalities are very difficult to discriminate the misfolded disease-causing proteins only. So that's why we developed the new therapeutics. So our therapeutics enables the specific elimination of only misfolded engaged proteins. And I will explain more details at the next slide. So we call it JUMP70 technologies. This JUMP70 technology is based on the designed fusion proteins named JUMP70 proteins. JUMP70 protein is composed of mainly two domains. The first domain is a protein folding activation domain. This protein folding activation domain binds to HSP70 chaperone proteins and activates HSP70. To make it specific to engaged proteins, protein folding activation domain is linked to targeting domains. Targeting domains can bind to disease-causing proteins. Right. So this JUMP70 protein is expressed in patient nerve cells by using gene therapies. And in the cells, the JUMP70 protein first binds to disease-causing proteins through targeting domains. In parallel, the protein folding activation domains of JUMP70 proteins brings HSP70 proteins to the complex to form ternary complex. So now the complex contains the JUMP70 proteins and the disease-causing proteins and the HSP70. The next step is especially interesting. Only when the bound and engaged disease-causing protein is misfolded, this disease-causing protein is transferred to HSP70 because HSP70 can recognize only misfolded proteins in the vicinity. And once disease-causing protein is transferred to HSP70, another function of protein folding activation domains works, which is activation of HSP70. Therefore, the consequence of disease-causing protein is defolding degradations by protein degradation systems associated with HSP70. Through this cycle, the JAMP70 protein is released from the complex, and now JAMP70 protein is free to engage another disease-causing proteins. So JAMP70 protein itself works catalytically. So this slide shows that how we design these fusion proteins. First, we identified the protein folding activation domains through in-house screening, and we have optimized the sequence, so we don't need to touch this sequence. So maybe we designed and engineer targeting domains. The targeting domain can be a peptide sequence, which is screened through a page display, or antibody-like sequence, such as single-chain variable fragments. So for instance, the Huntington disease is caused by a mutation in Huntington gene, and the translated mutant Huntington protein has expanded polyglutamine lipids. This expanded polyglutamine lipids renders Huntington proteins prone to a misfolding, which is believed to cause the Huntington disease. So the SOL-175 carries the binding sequence for polyglutamine lipid. And another program is for Parkinson's disease. And Parkinson's disease is characterized by Ruby bodies, and the major components of Ruby body is aggregated and misfolded alpha-synuclein. So SOL-368 carries the binding sequence for misfolded alpha-synuclein. Currently, we are developing this platform therapies in conjunction with gene therapies using AAV. So AAV is basically the genome of AAV is not integrated in the host genome, so it's less carcinogenic. Also, the AAV is less immunogenic compared to other vehicles such as lentivirus or adenovirus. And some of the serotypes of AAVs have a good tropism to nerve cells. And also, we tentatively selected AAV-RH10 because AAV-RH10 has a good tropism to nerve cells. And also, the AAV-RH10 can penetrate the blood-brain barrier. So in the future, it's feasible to develop these gene therapies with less invasive methods. Okay, so again, the JAMP70 technology can eliminate the misfolded engaged proteins. So this is a completely new modalities. And it works catalytically, so it's very potent. And it's very specific to a misfolded engaged proteins, owing to 2 levels of specificities. The first specificity is brought by targeting domains. Targeting domains engages misfolded disease-causing proteins. But even if targeting domains binds to properly folded disease-causing proteins, the bound folded disease-causing protein cannot be processed because HSP70 can be activated only for misfolded proteins. And the simple domain configuration allows us to design the new JAMP70 proteins for different target disease-causing proteins. So we can easily expand the disease indications. And also the genes encoding JAMP70 proteins are relatively small. Currently, the packaging capacity of AAV is around 4,500 base pair, but all of the genes we have now is less than 1,000 base pair. So it's very suitable to AAV-based gene therapies. And currently, we have 10 programs in our pipelines. And the most advanced one is SOL-257, of which indication is ALS. And the second leading program is Huntington disease, SOL-175. But today, I want to focus on SOL-257. Okay. And I don't think I need to explain this, but our target is TDP-43. The reason is the more than 95% of patients have a TDP-43 pathologies, and so we believe that the patients have two types of TDP-43. The one is, as I said, the good one, which is properly folded, but another one is bad one, which is misfolded and eventually causes neurodegeneration. So the difference, actually, most of the ALS patients have a TDP-43 pathologies without any mutations in TDP-43. So gene sequence itself of these two species is exactly same. The difference is just the structure. The bad one is misfolded. So we believe that our therapeutics is very suitable to TDP-43 targeted therapies. Our therapeutics can eliminate only misfolded NGJ proteins. So we designed our molecules for TDP-43. So the protein folding activation domains. Again, this sequence binds to and activates HSD70. This sequence is conjugated to a single chamber fragment, which preferentially binds to a misfolded TDP-43. And we named it SOL-257. First, we tested SOL-257 in cultured cells. So in these studies, we expressed many TDP-43 reporter constructs. In these studies, we expressed the type of TDP-43, which lacks the nuclear localization signals. So this reporter construct is dominantly localized in cytoplasma and eventually forms aggregates. When we stained with anti-TDP-43 antibodies, as you can see here, the aggregates is strongly immunoreactive to a phosphorylated TDP-43. And then we applied SOL-257. We expressed SOL-257, and we found that SOL-257 strongly suppressed the phosphorylation of TDP-43 aggregates. We also did some biochemical assays. So after cell lysis, the cell lysis were fractionated into a detergent of soluble fraction and insoluble fractions. And we did a Western blotting. So as you see here, actually, a second, okay. So most of the wild type TDP-43 is fractionated into soluble fractions. But when we express the TDP-43 delta NRS, some of them are also fractionated into insoluble fractions. And this fractionated TDP-43 delta NRS is also very phosphorylated. The expression of SOL-257 strongly suppressed the phosphorylation and also rescued the TDP-43 from insoluble fractions. So we also did so many cell-based assay, in vitro assays, and with these data, we decided to move forward to preclinical studies. First, we produced the AAV, and then we confirmed the expression from produced AAV using cultured cells. So we use primary mass cortical neurons and some other neuronal cell lines. After confirming the expression, we first injected the SOL-257 AAV into a wild-type mouse to confirm the safety and expression in vivo. And then we injected the SOL-257 AAV RH10 into one of the TDP-43 related mouse models. So this slide demonstrated that the safety of SOL-257 AAV. So we injected we injected SOL-257 into a wild type mouse through intrathecal or intracellular ventricularly. So both administrations show that the relatively safe. As you see here, we monitor the body weight to change, but there's no significant difference among the groups. So the blue one is control AAV-RH10, and the green one is SOL-257 AAV-RH10. And at the same time, we also injected SOL-175 for Huntington disease, but all of them show that there's no big difference, significant difference. And also we confirmed the expression. This slide shows that the expression in spinal cord. So after administration, we took the spinal cord and stained with antibodies. And the green one is signals from SOL-257, and the red one is from nerve cells markers. Right. So now we confirmed the safety and also expression. So we injected the SOL-257 into TDP-43 mouse models. We selected the RNLS8 mouse models. So some of the previous speakers also use these mouse models. So this mouse models expresses the mutant type of TDP-43 in the absence of doxycyclines. So this is a set of systems. The expressed protein is a TDP-43 delta NRS. So first, the mouse is fed with doxycycline-containing tube, but at 5 weeks, the doxycycline is removed from their food, and therefore the mice start to expressing the mutant type of TDP-43, and therefore they develop the TDP-43 pathology, such as mislocalization, aggregation, and phosphorylation, and gliosis, neurodegeneration, and eventually die within 5 to 6 weeks. So this is a very aggressive mouse models. The mice were assigned into 3 groups. The first group is control AAV was injected first, and but the mice were kept with doxycycline-containing tube, so they do not develop TDP-43 pathology. So this is a negative control. The group 2, the control AAV was injected, and at 5 weeks, doxycycline was removed from their food. And the third group is a treatment group. SOL-257 AAV-RH10 was injected. Okay. So first we monitored the body weight change. The group 1 is a black line, so they gained the body weight, but group 2 is the disease model mouse, and they start losing their body weight after doxycycline was removed. Very interestingly, the group 3 mouse, this is a treatment group. This mouse shows that the attenuation of body weight loss. And more surprisingly, when we monitored its survival, the group 1 sustained 100% survival. Of course, this doesn't develop the TDP-43 pathologies. But when we monitored group 2, the survival rate sharply drops after doxycycline was removed. And it eventually reached around 37%, 5 weeks after doxycycline was removed. But the group 3 mouse, this is a treatment group, they sustained 100% survivals. So unfortunately, we did not do animal behavioral studies in these studies. So now we are repeating these studies, and this new study is focusing on behavioral studies. We also we're gonna monitor the survival over 10 weeks. So again, so this is the preclinical development before clinical studies. So first stage is production, and the second stage is the in vivo expression studies, and the third stage is in vivo expression and durability studies. And then we are repeating the in vivo efficacy studies. And again, we are focusing on behavioral studies. This study was supported by ALS associations. And however, we know that the most of the ALS patients develop TDP-43 pathologies without any TDP-43 mutations. So we do want to test our SOL-257 in this background. So the mouse model that develop TDP-43 pathologies without the TDP-43 mutations. So we selected one of the such mouse models. We selected C9orf70 the mutation mouse models. These mouse models, the first the hexanucleotide was is injected. This mouse develops the dipeptide pathologies as long as TDP-43 pathologies. So this study has just started. So hopefully, in the future, we want to present this data at the future conference. And this study was also supported by ALS associations. Also, the the recent discovery is centered on TDP-43 splicing abilities, especially for STASMIN2. And because the cryptic exon is not conserved in mouse, so we do want to test SOL-257 in some kind of human like cell lines or human models. So another study, important study, is to use iPSC studies derived from ALS patients with TDP-43 mutations. So we are also doing the iPSC studies, and in these studies, we mainly focus on TDP-43 functions. The STASMIN2 expression or something like that. All right, so thank you so much for your attention, and I would be happy to answer any questions.
Thank you. Fascinating technology, fascinating results. We have a couple of questions from the chat, one from Sophia Bardale. Do you have histology data from the SOL-257 efficacy study in the transgenic TDP model?
Yes, actually today I didn't show that data, but we did that. And we see that SOL-257 suppressed some of the TDP-43 pathologies like phosphorylation or aggregation of TDP-43.
Thank you. And another question from the chat, Pavlina Konstantinova. Are you planning to inject mice later, so not immediately after birth, and monitor efficacy then? Would you expect enough efficacy in that model if you did that?
Well, so the answer is yes, and but not in these mouse models, because we know that we first did the expression in vivo expression studies, and now we know that the expression from injected AAV takes at least 3 or 4 weeks. Maximum expression reaches around 8 weeks, so it takes time. But using these mouse models, the disease is very progressive, and within 5 weeks, the mouse would die. So this wouldn't allow us to inject like at 5 weeks or 6 weeks or 7 weeks. Before the AAV expression, AAV is expressed, the mouse would die. So that's not good. So in another mouse models, so C9orf72 mouse models, we inject, we're gonna inject the SOL-257 AAV-based at 6 months. So at 6 months in these mouse models, the TDP-43 pathology has already emerged. So I think in these mouse models we can prove that the SOL-257 treatment efficacy.
Thank you. A follow-up on that question, though I'm not an expert with the model, but the Delta NLS you said is a docs off model. Couldn't you wait longer, treat the animals later in life, and then wait a little bit longer to withdraw the doxycycline to sort of activate the TDP expression?
I think yes, I think we can do that. And but this mouse model was developed at the University of Pennsylvania, and their protocol is the doxycycline is removed at 5 weeks. So we don't want to change the protocol, but some companies also provide the modified protocols, and in this protocol, with this protocol, the doxycycline concentration is just reduced, not completely removed. And this way, the TDP-43 pathologies it's not developed so quickly. So in this mouse model, probably it is possible to inject SOL-257 after the TDP-43 pathologies has emerged.
Well, thank you very much for, again, a fascinating talk. We'll be moving on.